This chapter's on the cell. It's chapter 3. So we're going to look at the different types of cells and what are the different organelles in the cells, what their function is. If we start by looking at, there's a lot in your book about the history. Don't worry about that. Okay, I'm more concerned that you learn what are the components of the cell versus all the back history of this guy did that and this one found this. And I love it when you read through one whole thing and then say, oh, well, so-and-so disproved that. The one I will mention is with Louis Pasteur, who's a Frenchman. We've talked about him in chapter 1 briefly. And just from the standpoint that he did disprove the theory of spontaneous generation way back in the 1700s up until he did stuff in the 1800s, people felt that spontaneous generation occurred, that you had a piece of meat out and then suddenly there were flies on there. Had nothing to do with the fact you left the meat out for 2 weeks. And now we don't think anything of it. We accept that, yes, cells come from pre-existing cells. One of the reasons why I'm mentioning his work, though, is with his experiment that disproved that theory of spontaneous generation. It's a very famous experiment. He made these what are known as swan neck flask. You can see in this picture here how it has like the neck of a swan. So the end is open. He made broth, sterilized the broth. This, with the swan, or that kind of S-shaped, any dirt or debris that could come through this open would settle in the bottom part here. But air could still get through, and this remains sterile. Part of the reason I'm mentioning that is that he had several of these flasks, and there's still one of them in one of the museums in France. One of his original flasks is still not contaminated, which to me, I just sort of find that fascinating. So that little bit of history there for you. But he did disprove it. So the cell theory says that, number one, the cell is the fundamental unit of life, and that cells come from pre-existing cells. They're not just going to magically appear somewhere. There has to be a previous cell for new cells to come from. The endosymbiotic theory is something that a lot of people have discussed and figured out. And what they're saying now is, and in this chapter we'll talk about the functions of both the mitochondria and the chloroplast. And a big question, they are found in eukaryotic cells. So plant cells, animal cells, fungal cells, and protista cells. They're not in bacteria. They are not in the archaeas. The mitochondria is responsible for making energy. The chloroplast carries out photosynthesis. The question, or why they've been looked at so much, is that both of these structures have their own DNA in it, which kind of makes you wonder, why is it there in the first place? And the structure of the DNA is similar to prokaryotic cells. It's circular. Eukaryotic cells, the DNA is linear. So even though they're in a eukaryotic cell, the structure of the DNA, it's like, it doesn't look right. It's circular instead of linear. Why is that? The way mitochondria and chloroplasts will reproduce is very similar to what is known as binary fission, which is the way bacteria divide. The ribosomes that are present inside both of these structures also is similar to the prokaryotic ribosomes. So everything, and you look at the genetic sequencing of it, is very similar to prokaryotic cells. So you have all these markers that are very similar to prokaryotic cells, and the big question is why? And so at the endosymbiotic theory, what most people feel now is that the mitochondria and the chloroplast, way back sometime, were actually bacteria. And they were engulfed or eaten by a eukaryotic cell. But they weren't killed inside the cell. And so it's a true symbiotic relationship where one, the mitochondria or the chloroplast, those original bacteria, those cells are inside the other eukaryotic cell. It became a benefit to both. That, let's say you look at the mitochondria, if it was a bacteria and it was engulfed, way back then, the environment on Earth was not what it is today. It was very harsh. And so, and even for us today, this would provide the mitochondria / bacteria with a relatively safe environment, a lot of protection. So that's the benefit to it. What was the benefit to the eukaryotic cell? It now has somebody inside of it who's going to make energy for it. And so it's a win-win for both. So that's what we mean when you hear about the endosymbiotic theory, is that mitochondria and chloroplasts, we feel, were bacteria that have now evolved in a symbiotic relationship with the eukaryotic cells. And that's all this diagram here is showing us, that you had these organisms that were engulfed and now they're symbiotic together. Germ theory. What this says is that diseases result from an infection of microorganisms. Throughout, as I said, your book goes through a lot of history on things. So a lot of people played roles in this. Semmelweis and Snow discovered the role of sanitation in preventing diseases, meaning if you have a clean water supply, that does wonders to help prevent infections. When we talk about individual diseases, you're going to see a whole lot of them. How is it transmitted? By ingestion of contaminated food or water. So water, that's why it's so important whenever there's any type of a natural disaster or war or things like that. Make sure that people have clean water, and that will help prevent disease outbreak on top of everything else you're dealing with. So Snow was very good with figuring out, be careful where you get your drinking water from. Maybe don't get it from right below a sewage plant. Maybe go upstream. And then Semmelweis was very good. Wash your hands between patients, which we now just take for granted. That was not always known. Joseph Lister, Louis Pasteur, and Robert Koch, they did a lot to determine what was the causative agent for specific diseases. And then Robert Koch came up with Koch's postulates. So when you look at cells, they're divided as one of two types. You have prokaryotic cells and eukaryotic cells. Prokaryotic cells are typically smaller. There is no nucleus. They do have DNA, but there is no nucleus. They have no membrane-bound organelles. So there's no mitochondria, there's no chloroplast. Eukaryotes are larger. Their DNA is found enclosed in a nucleus. A little extra protection there. And they do also have membrane-bound organelles, and these organelles have very specific functions. So like the mitochondria is to make ATP, which is energy. And just kind of a word, even though prokaryotes don't have these membrane-bound organelles, they can still carry out those functions. They just have a different way of doing it. So if we look at the prokaryotic cells, we're talking about those organisms that are in the domains archaea and bacteria. As I said, there's no nucleus, no membrane-bound organelles. The DNA, as I mentioned a little bit ago, is circular. It's in what's known as a nucleoid region. It's not a special containment area of the cell. It's just an area where the DNA tends to congregate. The ribosomes are 70S. S is a measurement used Svelberg for weight. They're smaller than the eukaryotic cells. Prokaryotic cells I often describe as, it's kind of, in this area, you'd be used to like a stew or a gumbo. Everything is just thrown in the pot here. So this is your DNA, you have the ribosomes here, but it's all just mixed together. In terms of the morphology, remember morphology refers to shape. Different terms that we use for bacteria. If it's spherical, it's a coccus. If it's rod-shaped, it's bacillus. If it's kind of a curved looking rod, kind of like a comma, it's what we call Vibrio. If it's oval, it's called Coxobacillus. If it's spiral, it's Spirillum. And if it's a long helical spiral, it's called Spirochete. And then we, in addition to morphology, we talk about the arrangement. And so we talk about, is it single? And you can do this for both cocci and bacilli. These are going to be the two most common shapes that you see are cocci and bacilli. Is it in pairs? Is it in smaller groups? Is it chains? With bacilli, is it a chain end to end like this? Or is it what we call palisade, where the long side is backed up to each other, kind of like making a fence. The cell wall. The cell wall is going to help maintain the structure. All cells have a cell membrane. Not everyone has a cell wall. With bacteria, most do have a cell wall. If you have a cell wall, it's going to be outside of the cell membrane. It's going to help maintain the osmotic pressure in there. We look at the chemical composition to help classify what kind of an organism it is because it's unique to that group. Plants have a cell wall that's made out of cellulose. Fungi have a cell wall that's made mostly out of chitin. For the prokaryotes, bacteria have a unique compound that's called peptidoglycan. The archaea have a cell wall, but it is not containing peptidoglycan. So that's a huge thing that you can use to tell the difference between the two, is run a chemical analysis of that cell wall. Tonicity is something that we talk about in most biology classes. And basically what you are doing is you're looking at the concentration of the solute or things that are dissolved inside a cell and comparing it to what is outside the cell. And why are you doing this? Because it's going to make a difference in which way water is going to flow. So an isotonic solution, iso means equal, and so that means that the concentration of the solute inside the cell is the same as outside the cell. So there's no net movement of water. And that's the way most organisms, that's the natural environment they're going to be in. If you have a hypertonic solution, hyper means more. And so that means that the solute concentration is greater outside the cell than inside the cell. And what's going to happen is water is going to move out of the cell. It's going to start to shrink. Now, having a cell wall is going to give a little bit extra protection than if it didn't, but you're still going to have water moving out of the cell. A hypotonic solution, hypo means less, so therefore the solution, the concentration of the solute of what's dissolved in the solution outside the cell is less than what's inside the cell. So now water is going to move into the cell, and water is going to keep moving in. If there's no cell wall, that cell's going to burst. Having a cell will help prevent the cell itself from bursting, but there may be a point of no return. So there are some bacteria that do not have a cell wall, and I said this is what's going to happen in the various types of tonicity situations. Is isotonic, the cell's going to be normal. In a hypertonic, the cell's going to shrivel because water is leaving, and in a hypertonic, water keeps moving in, and that cell is going to swell up and eventually burst. So you have to be aware of these things. Now, like I said, having a cell wall gives extra protection. So here's in the isotonic situation. The hypertonic, where water is moving out, see how this would be the cell membrane here. It's pulling away from the cell wall. It's going to help keep the cell from bursting, but you're going to have problems. And likewise, having the cell wall is helping to prevent the cell from bursting because of the influx of water. But it can only do so much. Still talking about prokaryotic cells, the nucleoid region is the area that has that DNA. The DNA is circular. It is haploid. Haploid meaning it only has one copy of every gene. It's not membrane bound in terms of there's no membrane around it. That's what we mean. Now, sometimes prokaryotic cells have a plasmid in them. The plasmid is not in the nucleoid region. It's going to be in a different area of the cell, just kind of hanging out there. What a plasmid is a small circular extra piece of DNA. And certainly in medical microbiology, why we're so concerned about plasmids is that oftentimes the genes for drug resistance, for antibiotic resistance, are on the plasmid. So that's why we're usually very concerned about it. The ribosomes are the site of where protein synthesis will occur. Ribosomes are made up of a special type of RNA called ribosomal RNA, which we abbreviate as rRNA, and proteins. There are 2 subunits, and they total 70S. So during protein synthesis, these 2 subunits, there's a small one and a larger one, they will come together. Yes, I know these numbers do not add up. Don't worry about that. Like I say, it has to do with a weight measurement. But for prokaryotic cells, it's going to be 70S. Inclusions. There's different types of inclusions that may or may not be found inside a prokaryotic cell. An inclusion just means it's something that's storing chemicals or extra nutrients. It may be storing starch, may store sulfur, may store gas or iron oxides. Endospores. Some cells produce endospores, not all of them do, but some will produce endospores. The purpose of an endospore is survival. It helps the bacteria to survive during very adverse or harsh environmental conditions. What happens is when a cell encounters very harsh environmental conditions, it will trigger the production of the endospore. The endospore, it's kind of like a dormant state. It will just kind of hang out there. The regular vegetative cell may die, but the endospore its purpose is survival, and so it's very hard to kill them. They just kind of hang there, and then when favorable conditions return, then the spores will germinate into new cells. So it's definitely a survival mechanism. So this is showing up here the DNA replicates. You've got this cell. It replicates the DNA so it can make the spore. Here's the progression. Here's the spore. This vegetative cell, the cell that was growing initially, dies due to the harsh conditions, but that endospore is released and it survives. This is from an electron microscope, and over here you can see swelling in some areas of these cells. We talked about the endospore stain last time, where you can see, like I said, they're very difficult to kill. So it's something you have to kind of keep in mind when you're talking about how do I sanitize things? Do I get the spores or not? And we'll talk about that later. The plasma membrane. So this is inside the cell wall. All cells have a plasma membrane. It is selectively permeable. That means it's not a free-for-all as to what can move in and out. Only certain things can move in, only certain things can move out. It is a fluid mosaic model, just meaning that the components of that membrane, they kind of move back and forth. I often describe it as sort of like looking at seaweed or kelp, and that, you know, with the waves, it just kind of back and forth. So it's not like it's cemented there in place of like in concrete and it can't move. It's composed of a phospholipid bilayer, and it has proteins that will be embedded in it. And so like these components, they kind of go back and forth a bit. Here are your phospholipids. It is a lipid type compound. Bilayer, so you have 2 layers of it. You do have things like cholesterol embedded to help stabilize. All of these larger compounds right here, these are all proteins that are embedded within that membrane. And those proteins are going to be unique depending on which organism you're talking about. So the proteins are not identical from one species to another. And like I said, it's going to be selective as to what can pass through. Movement across the membrane can either be passive or active. Passive means no energy is required. Why? Because you're moving in the direction things naturally want to move. You're moving what we call down a concentration gradient. Things naturally want to go from high concentration to low concentration. So if it's moving in that direction, it's passive. It doesn't require any energy. Active transport does require energy because you're going against the flow. It's like you're trying to paddle upstream. It's not the way you want to go. 3 different types of passive transport. There's simple diffusion, facilitated diffusion, and osmosis. Simple diffusion, just moving down the concentration gradient. You have a lot of these green structures on this side of the membrane, and so they're naturally going to want to flow in this direction from high to low concentration until equilibrium is reached. Doesn't need any help. Facilitate diffusion, all that means is you need a protein right here to help you move across. It may be because this phospholipid membrane is hydrophobic. It's a lipid. It doesn't like water-based or water-dissolvable compounds to pass through. So sometimes it's almost like going through a straw here. You need help. Osmosis just refers to basically the diffusion of water, the movement of water across the membrane. Active transport, you are going against that membrane. You've got a concentration gradient. You've got high concentration, low concentration. You're going against the flow. Probably the best example of this is what we call the sodium-potassium pump. And so here, if on the bottom, this is inside the cell, and on the top is outside the cell. So here's your plasma membrane. What the sodium-potassium pump is going to do, it's going to require energy. The green little, what are these, hexagons, are sodium. Sodium is higher outside the cell, but some of it has leaked in. In order for certain chemical processes to work, you need to have a concentration gradient. You don't want it to reach equilibrium. So what you're going to do is you are going to move sodium out. And so it naturally is wanting to flow in. But what you're going to do is you're going to pump, for each turn, you're going to pump 3 sodiums out and 2, the red is potassium, 2 potassiums back in. That's going to cost you some energy to do that because you're moving it against the flow. But you have to maintain that concentration gradient. You cannot let it reach equilibrium. If you do, the cell's going to die. So it's a case of, yeah, it's going to cost me some energy, but it's going to keep me alive. With the cell wall now, for bacteria, peptidoglycan is that compound I mentioned that is found in the cell wall. It's only found in cell walls of bacteria. There are structural differences in the cell wall between gram-positive and gram-negative bacteria. And this is going to help explain why they pick up the gram stain differently. It helps to explain why they respond differently to antibiotics, which is ultimately what you're concerned about from a medical standpoint. So the peptidoglycan, what is this thing? It's layers of alternating compounds. One of them is called N-acetylglucosamine, which we abbreviate as NAG. And the other compound is N-acetylmuronic acid, which we abbreviate as NAM. You'll find out in microbiology, we don't like writing out long words all the time, so we use abbreviations a lot. So you have alternating NAG with NAM, and you're going to have layers of this. And the layers are going to be connected by peptide bridges. It's going to look like an open mesh, or often described as looking like a lattice. And so here's basically what it looks like. So on a gram-positive, you have, the way they've done this diagram is the blue circles are NAG and those orange circles are NAM. So notice you've got alternating. So you've got just layers or lines of these. So you have here and here and again here. Now, what it doesn't exactly show is how are these all going to be connected with the layers underneath. You're going to have the little circles here are peptides that are helping to connect them together. This is gram-positive over here, gram-negative over here. So with the gram-positive bacteria, their cell wall has a very thick layer of peptidoglycan. So it's multiple layers of it. It's very thick. They have embedded in it what is known as teichoic acids. When you do a gram stain, it will stain purple. The gram-negatives, they have a very thin layer of peptidoglycan. In addition to that, on the outer side of the cell wall, they have another layer. They have what's known as a lipopolysaccharide membrane. So this membrane is not present in your gram-positives. One thing to keep in mind is that in this membrane, it does contain what's known as lipid A. This is an endotoxin. When this is released, it does trigger things such as a fever, inflammation, hemorrhage, can lead to shock. So you have to remember that. This is going to sound, I know, really weird. If you're treating somebody who has a really bad gram-negative infection, you don't want to kill all the gram-negative bacteria at once, because yes, you want to kill them to improve the infection, but you don't want to kill them all at once, because then you would be dumping all of this lipid A into the patient's body, which is going to send them into shock. And now you've got another problem on top of the original problem you had with the infection. So another reason of why it's important to know, does your patient have a gram-positive or a gram-negative? If they have a gram-positive infection, you don't have to worry about this. But if it's a gram-negative, and this is where it sounds really kind of weird, you're going to want to do more of like a controlled killing, like slow kill, not kill it all at once, because you don't want to send your patient into shock. A gram-negative bacteria will stain red with the gram stain. So on this diagram, it helps to show right here is the plasma membrane. Both your gram-positive and your gram-negative bacteria have that. So this is inside the cell. They both have the plasma membrane. Look right here with the peptidoglycan. You have multiple layers of it in the cell wall. These straight lines through here, those are the teichoic acid sticking up, helping to stabilize it. Versus over here, that's it, pretty much one layer of the peptidoglycan. And then you've got this outer membrane that also has the lipid A attached to it. So what happens is, see how this looks like a lattice work? When you do a gram stain, that crystal violet literally comes in, and it's in between all of this area, all in here. The iodine helps to stabilize it, and so that's why it's going to look purple. Versus over here, it just goes in this area pretty much, and here. But then what happens is when you do the step of adding the alcohol to this step, what's going to happen then is alcohol will destroy lipids. This is a lipopolysaccharide outer membrane. All of this gets ripped off, which then means if you rip this off, you destroyed the lattice work, and there goes the purple dye. So this disappears, this disappears, this disappears. This is completely gone now. So now when you add the saffron red, these spots are empty. And so the saffron red dye can sit there. And that's why your gram positives look purple and your gram negatives will look red. It has to do with the fundamental structural differences between the cell wall. A lot of it's based on peptidoglycan and then that lipopolysaccharide layer. Glycocalyces. These are structures that are on the outside of the cell wall. They may or may not be there. They allow attachment to surfaces. They're involved with helping to form biofilms, their protection. This includes things like a slimy layer and a capsule. So you won't necessarily have both. You may not have either one. Slime layer is very loosely attached, so it can be easily washed off. The capsule is much more organized. It's tightly held to the cell. The capsule, we have found, does help increase virulence. That there are times you may have a bacteria. One of them has a capsule, one does not. If it does not have a capsule, it's not going to be pathogenic. It's not going to make you sick. If it has a capsule, yeah, it's going to probably make you sick now. So all is showing that you've got your innermost is your plasma membrane. Then outside of that is the cell wall. Outside of that would be the capsule. If you look on this microgram, you can see that there's kind of a whitish color surrounding some of these cells. That's because it's the capsule. So the cell itself looks kind of purplish here, and then the background is a very pale purple, but that whitish surrounding the cell, that's the capsule. There may be appendages. Once again, not all cells have this. Fimbriae helps attach to surfaces. Pili, number one, fimbriae usually are completely surrounding the cell. Pili, they will connect, it's usually just one tube, connects two compatible, they're only found in gram-negative bacteria. Pili, if they produce pili, it has to be, number one, it has to be gram-negative, and the cell it attaches to has to be compatible. There has to be the same species, and there have to be some other markers to make it compatible. And so what the importance of the pili is it allows for a transfer of DNA between the two different cells. And then another appendage is flagella that helps to provide motility. Flagella, your book, once again, this chapter goes into a lot of detail. All you need to know is that it is composed of a basal body, a hook, and the filament. So here's a picture of the pili, how you have the connection between the two cells, and DNA can pass between this. With the flagella, like I said, you just need to be familiar with the filament, which is this long structure here. So here's the filament. Here's the hook, which helps to stabilize it within the, allows it to turn and stabilize it along with the basal body, which is this whole structure right here, that secures it within the cell, kind of anchors it in. That's all you need to know in terms of the structure of it. There are different types of flagella. This is important from the standpoint of helping us to identify and characterize different bacteria. Like I say, number one, not all bacteria have flagella. Those that do, we look at what's the location and how many of them. If there is just one flagella, and obviously it's just at one end of the cell, it's called monotrichous. Amphitrichous is where you have a flagella at each end of the cell. So oftentimes it's bacillus or rod-shaped, and so at each end there's flagella. It may be a single one or maybe a little tuft coming. Lophotrichous is where you have only at one end of the cell the flagella. The difference between the mono and the loph is that the mono has just one, but the lopho has several flagella. They're all only at one end of the cell. And paratrichous is where it's surrounding the entire cell. So here's the monotrichous, just one. Amphi, where they're at both ends. Lopho, where you've got that tuft of them. And paratrichous, where they're all over. So if a cell has flagella, then characteristically it's one of these types. It's not like, oh, today I'm going to be monotrichous, but tomorrow I'm going to be paratrichous. No. You see, say, a particular cell, if it's paratrichous, it's always going to be paratrichous. If it's monotrichous, it's always going to be monotrichous. So that's why you can use it to help in identification purposes. The flagella does help with motility of the cell. Cells move in what we call a run and tumble. They always say they never took geometry to know that the shortest distance between two spots is a straight line. They don't know that. So what happens when you watch them, it's kind of interesting. If you have some bacteria here and then it needs to go here. Like I said, they never took geometry to know that you would just go a straight line between these. No, they run and tumble. It's like they start here and they start going, oh, I want to go up there. And then sometimes they kind of roll around and tumble. Oh, let me go see what's over here. And oh, let's see what's over here. Oh, if it's me, it's going to be like, oh, chocolate. Oh, I want some Pepsi now. Let me go over here. And we're going to go around over here. And oh, we're going to kind of circle around. And oh, what was it I want? Oh, that's right. Yes, I want it to go. Oh, that's what I want. It's not a straight line. Like I said, they never took geometry. But it's kind of cool to watch them sometimes. Or annoying if you're trying to track them. But that's how they move. So this is just showing how they will use the flagella to help them move around. Now, your eukaryotic cells. In microbiology, we were talking about things like your fungi, your algae, some of your protozoans, your protistas, all of those. What are the differences? Well, number one, they have a membrane-bound nucleus, and the DNA is going to be inside that nucleus. The DNA, normally you have multiple chromosomes, not just one, there's multiple, and they are linear. The cell also is going to contain various membrane-bound organelles. It's usually larger than your prokaryotic ones. Cell division is going to occur by mitosis or meiosis. Ribosomes are 80S, so they're a little bit larger than the 70S that we saw with the prokaryotic ones. Some of the cells will have a cell wall, others do not. Very, very diverse morphologies, so it's not a standard shape to them. So here's kind of your representative eukaryotic organism. May or may not have a cell wall. It does have the plasma or cell membrane around it. Over here in the center right here is your nucleus that contains that DNA. But you have things like mitochondria. You have endoplasmic reticulums, Golgi apparatus, which we'll talk about. So you have all these different little organelles. One advantage of the organelles is that within the cell, you have these membrane-bound organelles, so they have their own membrane, so that inside the organelle, the environment might be slightly different than the rest of the cell, and that can optimize the conditions for whatever that organelle is supposed to do. Maybe it's supposed to modify proteins. And so you can have differences, kind of slight differences, they're usually not major, but you have differences in the environment within the cell to maximize, like I say, the various functions. Sometimes I give the analogy, it's kind of like my house in that years ago when my kids were still living at home, let's say when my son was playing sports in high school, he ran track, he played soccer, and we'll just say that his room, to put this politely, the atmosphere in his room was a little different than the rest of the room, in terms of maybe smell, in terms of he had stuff maybe thrown around. He was a typical high school athlete kid, run in, throw down his soccer cleats or whatever. There was a while there I remember when he has his taste in music. It is not country. For a while there, my oldest daughter was really like country music, so in her room she would have country music playing. That is not what my son listens to. He prefers more rock, metal, that type. And then my youngest, who's several years younger than the two older ones, was into the Disney Frozen. And so each of them in their rooms, number one, the music was very different. The toys or whatever, they were computer stuff, was very different. The environment, the decoration was very different. It's kind of like that in a cell, and that each of the organelles has its own environment, but it all works together to maximize the functioning of the cell. The nucleus, this is the control center for the cell. With my analogy of my house, I used to wish or would be in denial and say that I was the nucleus because I felt that I should be the control center. Sometimes it wasn't always true, but in the nucleus, it contains the DNA. DNA is double-stranded. Most books will tell you that DNA is always double-stranded. I'll just tell you right now, when we get to viruses, you will find there are some viruses that are single-stranded DNA. There is a nuclear membrane. Some cells have more than one nucleus. We do see this with fungi sometimes. What we call the cenocytic or cenocytes just means they have multiple nuclei. There's certain fungi are really strange in the life cycles that they go through. The nucleolus is within the nucleus. This is the site of ribosomal RNA synthesis. This is what I mentioned is one of the components of the ribosome. So the nucleolus, if you look at a cell, oftentimes, like in this drawing here is the nucleus, it's going to be a darker. When you look at it under a microscope, sometimes you can see the nucleolus. It looks like a darker circle within the nucleus. The ribosomes, the site of protein synthesis, same as what we saw with the prokaryotes. However, the subunits there's two subunits, just like what we saw with the prokaryote. However, instead of a 30 and a 50, there are 40S and a 60, and instead of a total of 70, it's a little bit bigger. It's an 80S. Now, the 70S ribosomes are found in the mitochondria and the chloroplast. This, I'm just going to say, why I'm stressing it, is that it becomes really important if you're trying to develop antibiotics to be effective against killing, say, bacteria that are prokaryotes. If you're trying to develop new ones, synthesize new ones, it would make sense that you look for differences between the prokaryotic cell versus our cells. Animal cells, we are eukaryotic cells. So our cells have the 80S ribosome. So look for the differences. That's why a lot of antibiotics, they're made to interfere with protein synthesis. They're made to go against the 70S. Because that's what's in the bacteria, not in our cells. However, you have to keep in mind, if you're taking an antibiotic that the way its mode of action, the way it works is against the 70S ribosome. You can't take that for long term, because with long term, then remember, you do have 70S ribosomes. As animals, we do have mitochondria. We don't have chloroplasts, but we have the mitochondria. So over long term, it could cause some problems. That's just the way you're looking at these differences. Once again, ribosomes, there's two different types of ribosomes. There's what we call bound ribosomes and free ribosomes. Free ribosomes, they're just circulating freely in the cytoplasm. The proteins that are made there are usually going to be used within the cell itself. The bound ribosomes, they are attached to what is known as the rough endoplasmic reticulum. Most of the proteins made there will be exported out of the cell. So here's your endoplasmic reticulum. There's two types. There's the rough and the smooth. And endoplasmic reticulum we just tend to abbreviate as ER. The rough ER has the ribosomes attached to it, so the purpose is you're making proteins there. Like I said, those are usually going to be exported out of the cell. The smooth ER, which does not have ribosomes, if you look at a picture of it, it doesn't have any little dots on it or anything. The purpose of the smooth ER is for things like detoxification, for producing lipids and carbohydrates. The Golgi apparatus, this is for protein and lipid modification. So oftentimes you make a protein, then you send it to the Golgi apparatus. When we talk about modifications, maybe you're sticking a lipid on it, maybe you're sticking a sugar on the end of it, you're modifying it somehow, and then you send it out. Lysosomes, this is like the recycling center. Lysosomes are little vesicles that have highly digestive enzymes inside of them, and so when a cell engulfs, like say one of your white blood cells have a lot of lysosomes in them, so that when they engulf a foreign cell, it fuses with the lysosomes and basically it breaks it down, it chews it up. Vesicles are going to be like little containers that are holding other things. So in this drawing, here's the rough ER. You can see it has those little red dots on it indicating the ribosomes attached. The smooth ER, nothing is attached to it. Here's your Golgi apparatus. The Golgi apparatus, the vesicles, oftentimes what happens is you make a protein on the rough ER. It breaks off, is enclosed inside a little vesicle, inside this little sac, which then moves to the Golgi apparatus, fuses with it. It's going to pass through these layers. I describe the Golgi apparatus as kind of looking like a stack of pancakes if you look at it. So it's going to, this vesicle with the protein is going to fuse to the top, gradually move down, just like your syrup dripping off. Once the modification has been done, then here's the vesicle here with the modified protein in it, and this is going to move towards the cell membrane and be exported from the cell. The cytoskeleton is for structural support. It helps to transport material intracellularly. It helps to anchor those organelles. It includes several different types: microfilaments, intermediate filaments, microtubules. The centrosomes will form spindle fibers during mitosis and meiosis. The mitochondria that we've mentioned several different times is the powerhouse of the cell. This is where most of the ATP is going to be produced. ATP, think of as energy. It is actually has a double membrane. Inside the inner matrix contains this mitochondrial DNA, which is circular, and that's also where the 70S ribosomes are. So here, as you can see, both in the electron micrograph of it, as well as the schematic drawing over here, that it is a double membrane. Just keep in mind, some of you may have had this from A&P. Whenever you see folding like this on this inner membrane, we call it invaginations, but whenever you see all of this inner folding like this, what that is doing is increasing the surface area than if you had just like the outer membrane, just a straight membrane. If you're increasing the surface area, that's telling you that there's probably something very important that is occurring there. But if you're looking like, why is it so kind of jagged? Like, it's increasing the surface area. Chloroplast, the site of photosynthesis. Remember, we're still talking about eukaryotic cells. What happens with photosynthesis is you're taking solar energy to produce glucose and oxygen. So this plasma membrane that all cells have, once again, it's a phospholipid bilayer. I've mentioned that you have embedded proteins in there. Integral proteins just means that it extends the entire width of the membrane. Peripheral is just on the edge. It's selectively permeable. Once again, only certain things are allowed in and out. Fluid mosaic model means that there is some slight movement with it. And as I mentioned before, cholesterol helps to stabilize it. So here is a drawing of your plasma membrane again. So you can see the bilayer. These little structures here is the cholesterol, the larger. Here's an integral protein because it goes the whole way, which apparently I can't draw that very well. There's an integral. This would be a peripheral protein because it does not extend all the way through. In terms of membrane transport for large substances, we talked about small things with passive and active transport. For large substances, sometimes things are just too big to pass through. So endocytosis is you're moving substances into the cell. Phagocytosis, you're moving solid things. So it's often referred to as cell eating. Penocytosis, you're moving liquid. It's often referred to as cell drinking. And receptor-mediated endocytosis, your binding is very specific as to what the substance is that you're bringing into the cell. Exocytosis, now you're moving the substances out of the cell. Oftentimes it's waste products. So phagocytosis, once again, here's a large particle that's too big to pass through the membrane. So you kind of just go around and surround it and bring it in a vesicle. Same thing with liquid for peno. Here, it's receptor mediated. It's dependent on what can bind. Binding of a substance to these receptors is what triggers the enfolding. So the cell wall with eukaryotes, plants, the main structural compound is going to be cellulose. Cellulose is actually the most abundant carbon compound on Earth. Fungi, it's going to be a little bit of cellulose, but mostly chitin. Chitin is similar to, that's what makes it like the hard shell, exoskeleton shell on insects, things like that. Beetles, things like that. Animals do not have a cell wall, but there's an extracellular matrix, some extra gunk outside of the cell, but it's not a cell wall. Proteins, it depends on the species. Proteins is a huge kingdom, lots of diversity. It's basically the catch-all of where, if you don't know where to put something, you know, it's not a plant, it's not a fungus, and it's not an animal. Throw it in as the protist. So there's a wide diversity. So it depends on the species that you're talking about as to whether or not they even have a cell wall. Most of them, if they have a cell wall, it's usually going to be made up of calcium carbonate. And then algae, it's usually going to be cellulose and pectin, but it's going to also be dependent on the species. Eukaryotic cells may have appendages as well, flagella to help with locomotion. It helps to move the cell through an environment. And then cilia are going to be appendages that are shorter than flagella. Usually if you have flagella, there's just one. Cilia are much more numerous. They're much shorter than flagella. And in this case, flagella moves the cell through the environment. Cilia, the cell stays still, but you're moving like fluid or material across the surface of the cell. So the cell stays still, but cilia allows you, the movement of it moves things across the surface of the cell. So that is just kind of a summary of comparisons of your prokaryotic cells versus your eukaryotic cells.